Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on Earth, and that's biology. So here we are in a very new series that's called Highlight Series. So here we're going to talk about the highlights of important chapters that in biology students feel tough. So we have started off with photosynthetic pathways. So this is highlights. I will just give you what to be remembered. So where you face a problem, and we'll finally summarize it. So that is what the highlight series is all about. So weekly, twice or thrice, you might have the highlight series, and the remaining day we will solve sample papers. So the last 1 month I will do one shot, not before that. All right. So we will have mentee coming in between, so don't worry. So highlight series. So we are starting with photosynthetic pathways part 1. So as you can see, photosynthetic pathways, so it involves your C3, C4, CAM and your C2 cycle. So this is where students actually feel a bit tough, right? Ma'am, C3, ma'am, Hatch and Slack pathway. So they give me all kind of reasons and suggestions to forget this topic, but no, that's not happening. So we have to do this topic. So this is the most important topic, I would say, from plant physiology. So you guys have to go through this topic. So that is why I thought I will do a highlights where I'll make you understand how it is happening and what are the points you should remember in each pathway. Right, so here we go. So today it is part 1, so I'm not going to complete the entire pathway, but let's see what I have in store for you. So under photosynthetic pathway, we have the C3 or the Calvin cycle, right? So under the C4 pathway, next we have the C4 pathway or the Hatch and Slack pathway, right? So we have the C3, the C4, right? So we have the C3 plants and the C4 plants. Next we have the C2 cycle, which is also known as photorespiration. The C3 cycle is also known as the Calvin cycle. The C4 cycle is also known as the Hatch and Slack pathway, because these are the scientists who discovered it. And then C2 cycle is also known as photorespiration. You're going to see why. So the last one that I want to explain to you is the CAM plants. So these are the 4 things that is very important from this chapter, that is your photosynthetic pathways, right? So today we'll talk about your C3 pathway and the C4 pathway. So C3 pathway, which is also called as the Calvin cycle. So C4 pathway, which is also called as the Hatch and Slack pathway. All right, yes. So we start with the C3 pathway. So what is that you have to understand in the C3 pathway is what I'm going to tell you. So right after the process of photosynthesis, so you know your photosynthesis is divided into 2 reactions. So one is light reaction, the other one is dark reaction. So light reaction is called so because it happens in the presence of light, right? And the end products of light reaction are 3. So what are the end products of your light reaction? So the end products of your light reaction include your oxygen, your ATP and your NADPH. So these are the net products of your light reaction. That is, it happens in the presence of light. So we make use of the photosystem 1 and the photosystem 2 for these end products, right? So the oxygen, so what happens to this oxygen is that it moves into mitochondria for the process of respiration. So what happens to this ATP and NADPH is that it is used in dark reaction. Your dark reaction is also known as your biosynthetic reactions. So what is this biosynthetic reaction? So that is where your carbon dioxide is fixed and converted into glucose, right? So that is where the enzymes present in the stroma help in the formation of a sugar. So the sugar in photosynthesis is your glucose, right? So this process of dark reaction happens in the stroma of the chloroplast. Right? So how this biosynthetic equation or how this biosynthetic phase happens? So how does the carbon dioxide get converted into a 6-carbon sugar molecule? So there we came across the C3 pathway. Right? So this ATP and NADPH are being used in the biosynthetic pathway, and this biosynthetic phase happens in the stroma of the chloroplast. So the cytoplasm of the chloroplast is called the stroma. So the first carbon dioxide fixation product, so the equation of photosynthesis is this. So you have carbon dioxide plus water, which gives you C6H12 O6 plus oxygen. So the oxygen is already formed from the light reaction. So this is a non-stoichiometric equation, meaning I'm not writing it in a balanced form, right? So carbon dioxide should be fixed to give you this 6-carbon molecule, that is glucose, right? So in the process of this conversion, the first stable or the intermediate product form is a 3-carbon compound. So that is why it is called as the C3 cycle. And the scientist who was behind all this is your Kelvin. So hence it came to be known as your Kelvin cycle. Alright, clear? Alright. So this Kelvin cycle or the C3 cycle, it occurs in all photosynthetic plants. Okay, so it especially occurs in the mesophyll cells. So you students always have a confusion between bundle sheath cells and mesophyll cells. So C3 happens in all photosynthetic plants, be it a C4 plant, be it C2 or CAM plant. C3 happens in all photosynthetic plants. All green plants that undergo photosynthesis has your Kelvin cycle happening, right? So in your C3, generally you have 2 types of plants, C3 and C4. Even in C4 plants, C3 cycle will happen. But the twist is, in the original C3 plant, this will happen. The process of this fixation will happen inside a mesophyll cell. Hi W, yes, a very happy good evening. Thank you for joining my session today. So please don't forget to like, share and subscribe. So what happens is this C3 cycle, in case of the C3 plant, happens in the mesophyll cell. So far what you have to remember? You have to remember Kelvin cycle is a part of the dark reaction, and this occurs in all green plants that undergoes photosynthesis, and the C3 plants exclusively it happens in the mesophyll cells. So mesophyll cells are the ground tissues, right? So which is neither vascular, which is neither epidermis, all right? Right. So this C3 cycle happens or occurs in 3 steps. What are the 3 steps we are going to see? The first step is carboxylation. The carbon dioxide is going to be accepted. So if I have gold if I have say gold bars with me, and if I have to buy something else, I have to do it in terms of cash. So I take the gold with me, give it to the person who will in turn give me cash, and I in turn purchase something, right? So similarly, someone has to accept my carbon dioxide here. So who is going to accept my carbon dioxide? So that is what we are going to see in carboxylation step. So be whoever it is, carbon dioxide has 1 carbon. Now it has to be converted into 6 carbons, so it depends on the acceptor, and then it has to be reduced into the C6H12O6 molecule, that is glucose. That is what will happen in reduction. So then the last step is regeneration. So we have read biomolecules. So if you remember how the enzyme substrate complex works, the enzyme is always coming back, or the enzyme comes out at the end of the reaction. An enzyme is never getting utilized in an enzyme catalyzed reaction, right? Enzyme always comes out. So much in the same way, whoever accepts carbon dioxide in the first step of carboxylation has to be regenerated in the 3rd step. So that is how the process of Kelvin cycle happens. Alright. So if you look, this is a straightforward diagram from your textbook, and I want you to go through all the diagrams from the textbook very carefully. So if you see, the first step is carboxylation. So carbon dioxide for the process of photosynthesis comes from the atmosphere, right? So the carbon dioxide comes, and the primary acceptor of carbon dioxide is ribulose 1,5-bisphosphate. So in the C3 cycle, the primary acceptor of carbon dioxide is your RuBP. So this RuBP is a 5-carbon compound. RuBP stands for ribulose 1,5-bisphosphate. Fine. So this is what is accepting your carbon dioxide. When this accepts your carbon dioxide, your first stable compound is formed. So I am writing the first step in C3 cycle. So the carbon dioxide is accepted by RuBP. This is a 5-carbon compound. When this accepts carbon dioxide, it forms 2 molecules of phosphoglyceric acid. This is a 3-carbon compound. So you have 2 molecules of 3 PGA. So totally 6 carbon is here. But this happens in the presence of an enzyme. So that enzyme is called as Rubisco. Okay. So what is the full form of Rubisco? So again, rubulose 1,5-bisphosphate carboxylase and oxygenase. Fine? Yes. So when this happens, this is your first step of carboxylation. This is step 1, that is carboxylation. Fine. So now we have step 2. So this 2 molecules of PGA, that is phosphoglyceric acid, which is 3-carbon compound, is reduced in successive steps to form your glucose. That is what is written as sucrose or starch. It is forming your glucose, right? So I write C6H12O6 for your understanding. So this process is your second step, that is reduction. But this step requires energy, the energy that was produced in the light reaction. What was the energy that was produced in the light reaction? ATP and NADPH, right? So here we are going to utilize 2 molecules of ATP to form 2 molecules of ADP plus 2 molecules of PI. Plus we also need 2 molecules of NADPH to form 2 molecules of NAD plus 2H plus. So how many ATP is being utilized? 2. How many NADH? 2. Right? So the last step here is regeneration. So this RuBP has to be regenerated. So the RuBP here is regenerated. So this regeneration of RuBP requires 1 ATP. So this is step 3, that is your regeneration. Fine. So that is what is shown here. So RuBP is accepts the carbon dioxide. This is carboxylation. It forms your phosphoglyceric acid molecule. This is reduced. When it is reduced, ATP and NADPH are being utilized. So it forms your ADP and NADP plus, and it finally reduces to give you your glucose or your sucrose or your starch, and then your RuBP is regenerated, for which again you need 1 molecule of ATP. So this is your C3 cycle. Alright? Right. So what is the energy consumed? So in the step of reduction, you have 2 ATP for phosphorylation and you have 2 NADH, which is used for the reduction step. And then you have 1 molecule of ATP for the regeneration of RuBP. But understand the equation of photosynthesis is that you have 6 molecules of carbon dioxide. So when you write a balanced equation, it is 6 molecules of carbon dioxide plus 6 molecules of water, which gives you C6H12O6 plus 6 molecules of oxygen. So all this energy consumed is for 1 molecule of carbon dioxide. So 1 molecule of carbon dioxide totally, I need 3 ATP plus 2 NADPH. So now for 6 molecules of carbon dioxide, it would be 18 ATP plus 12 NADPH. Right? Yes. So here we go. So the total energy gain for 6 molecules of carbon dioxide, 18 ATP has to be given inside. So what comes out is your 18 ATP. Then you give 12 NADPH and 12 NADP comes out. So now we are finally done with your C3 cycle. So I have tabulated the points to remember. First of all, your C3 cycle is also called as your Calvin cycle. And this Calvin cycle occurs in all photosynthetic plants. Be it C3 plants, be it C4 plants, it happens in all green plants that undergo photosynthesis. Right? Primary acceptor. Who is the primary acceptor of carbon dioxide here? RUBP. That is rubellose 1,5-bisphosphate. This is a 5-carbon compound. Right? So what is the enzyme which helps in this conversion? So carbon dioxide plus RUBP, it gives you 2 molecules of PGA. So here the enzyme is Rubisco. So this enzyme you should remember, rubellose bisphosphate carboxylase and oxygenase, right? So who is the first stable product? PGA, phosphoglyceric acid. So this is a 3-carbon compound, so that is why the C3 cycle. So where is the location? Where it is happening in C3 cycle? In the mesophyll cells. What is the energy consumed? 18 ATP plus 12 NADH for 6 molecules of carbon dioxide. So your C3 cycle is clear. So the final touch is done. No more doubt in C3 cycle. Can we go to the next one, that is the C4 cycle? Yes. W, have you understood the C3 cycle? So can we move forward with the C4 cycle? Yes. Alright, so we move. So we are coming to the C4 cycle. Ma'am, if you say C3 cycle happens in all photosynthetic plants, then what is the C4 cycle? So to be precise, not all plants can grow in normal temperature. There are some plants, so we have different ecosystems, right? We have tropical rainforest, we have deciduous forest, we have snow-laden mountains, then we have desert conditions. So towards the dry tropical area, whichever plant survives, they belong to the C4 category. Ashu, good evening. Yes, so you're saying good morning. Okay, a very happy good evening. Thank you so much for joining my session, Ashu. So please don't forget to like, share and subscribe. So we are talking about the C4 cycle. Alright, so this C4 plants are the plants that live in tropical dry regions. Okay. First carbon dioxide fixation product is a 4-compound oxaloacetic acid. Just like in the C3 cycle, someone has to accept your carbon dioxide and form a stable product, right? So here also someone is accepting the carbon dioxide, but the first stable product that is formed is a 4-carbon compound. So that 4-carbon compound was your oxaloacetic acid, and hence it is called as the C4 plants. That is the first difference. So in C3 plants, the primary stable product is a 3-carbon compound, that is PGA. Here it is a 4-carbon compound, that is your oxaloacetic acid. Alright, yes. But the main biosynthetic pathway is the C3 pathway. So this is where I am here to confuse you. So what is the biosynthetic pathway? The dark phase. So what happens in dark phase? Glucose is produced. So though the first stable product is oxaloacetic acid, the process by which glucose is produced is the C3 pathway. So in C4 plants, first the C4 pathway happens, then the C3 pathway happens. Alright? Yes? 4C, 5C. Okay, so 4C is 4-carbon. So when I say glucose, so glucose has 6 carbons, right? 3, 6. So that's why we say it is 6 carbon. When I say 5 carbon, it has 5 carbon backbone. So all you have to remember is RUBP is a 5 carbon compound, right? And then your PGA, phosphoglyceric acid, is a 3 carbon compound. Remember, it has 3 words. Here it is 3 compound, okay? But oxaloacetic acid, it is a bit heavy. So remember, it is 4 carbon compound. So this is all that you have to remember. Don't worry about anything else. Okay, W? Fine. So if C3 pathway is the main photosynthetic pathway, so how and where your C4 plants are special? There should be some difference, right? So that is why the first stable product there is your oxaloacetic acid, and in case of C3 plants, it is your PGA. So where is the difference? What is so special? Alright, so here we see. So what is so special in C4 plants? First of all, they have a special leaf anatomy. What is the leaf anatomy? The internal arrangement of the leaf. That is very different compared to a C3 plant. Alright. The next one is these plants, the C4 plants, can tolerate high temperature. So when I said they live in tropical dry regions, it is understood. Dry means you have high temperature and less of water, right? So they can tolerate extremely high temperatures. The third one is if they are in a high temperature, which means more sunlight is falling. So more sunlight means they are able to respond to high light intensities. So at high light intensity, the photosynthetic rate increases only up to a certain point, but they still respond to high light intensities. The fourth one is they do not have photorespiration. That is C2 cycle. As of now, keep this in check. I will explain you when I am explaining C2 cycle. So just leave off this point. The last point is they have greater productivity of biomass. So what do I mean? So biomass is the living tissue. So the efficiency of the C4 plants is much higher. So that is why they are special. Okay? Yes? Halophiles is salt tolerating plants. So halophiles, they can live near the marshy areas where they have more amount of water. So they need not be C4 plants. Okay, W? Yes? Okay. So why your C4 plants are special? Because they have a special anatomy, they have high temperature, they respond to high light intensity, they do not have the C2 cycle, and they have greater productivity or efficiency, right? So what is that special anatomy that makes the C4 cycle the best, right? So we will look into it. So you have large cells around the vascular bundle of the C4 plants. So when you talk about the structure of the leaf in anatomy, yeah, you're right, W, you're absolutely right. So when you talk about the anatomy of the leaf, the monocot leaf is called as the isobilateral leaf. Your dicot is called the dorsiventral leaf. So how many layers of arrangement you have in the leaf? The first one is the epidermis. Always the outermost layer is the epidermis. So in case of leaf, the second layer is your mesophyll. The third one is the vascular bundle, right? So vascular bundle includes your xylem and phloem. They say your xylem and phloem is surrounded by large cells. These cells are called as the bundle sheath cells. Suppose I have the xylem and the phloem here. So they are surrounded by sheaths that is called as the bundle sheath cells, like this. The xylem and phloem, which is the vascular bundle, they are surrounded by large cells that is called as bundle sheath cells in case of your C4 plants. Alright. So this type of arrangement is called as, okay, so this gives an appearance that is Kranz anatomy. So Kranz means wreath. What is that whorl? So suppose if you're going to give a bouquet to someone, what shape it is? So it can be this shape, right? So for a ornamental purpose, you arrange the flowers in this pattern. In much similar pattern, your crane's anatomy looks. This is how your crane's anatomy looks. If you see, the center is your vascular bundle. So your vascular bundle includes your xylem and your phloem. So can you see it is surrounded by huge cell, that is your bundle sheath cells. Surrounding the bundle sheath cells, you have the mesophyll cells. So bundle sheath cells and mesophyll cells lie close to each other. So this mesophyll cells form a structure that is known as a wreath-like structure. So your crans means this wreath-like arrangement. Clear? This is called as crane's anatomy. Which one? The mesophyll cells arranged around the vascular bundle. Clear? Now did you understand what is crane's anatomy? Yes? Right. So this is how it is arranged. So bundle sheath, vascular bundle, bundle sheath, then you have the mesophyll cells. So bundle sheath cells form several layers around the vascular bundle. So sorghum is your maize plant. Okay, it's a type of C4 plant which has this crane's anatomy. Alright. So this bundle sheath cells, they form several layers around the vascular bundle. They are not one layer, but they are many layers thick. Alright. So you have large number of chloroplasts here because it is several layers thick, so it can accommodate large layers of chloroplast. So this walls are impervious to gaseous exchange. Which one? The bundle sheath walls, they do not allow easy gas exchange to happen. Right? So there are no intercellular spaces. If you have seen the figure, they are very, very closely arranged. There is no intercellular space, right? So if you look here again, so there is no gap, right? Everything is arranged compactly. And within this space, there is no gas exchange. It is very, very thick. That is what they are saying. So what are the examples? Oh, okay, so one is maize, the other one is sorghum. Sorry, sorghum is a corn type plant, okay? So these are the two examples that is mentioned in your NCERT for your C4 plants. Fine? Okay. So now we move forward. How does the C4 cycle happen? C4 cycle first happens in the mesophyll cell. Second, it happens in the bundle sheath cell. Mesophyll cells and bundle sheath cells are very close by. So first it happens on the outer layer, then it moves to the inner layer. Clear? Yes? Okay. So first of all, what happens? Here we have to see who is the primary acceptor. So first of all, carbon dioxide comes from the atmosphere. It is accepted by a compound called as PEP. PEP stands for phosphoenol pyruvate. So this PEP is a 3-letter word. So this is also a 3-carbon compound. Okay. So this accepts your carbon dioxide and forms the first stable compound, that is your oxaloacetic acid, which is your 4-carbon, because of which it is called as the C4 cycle. The enzyme that is used here is called as peptidase. So what does this mean? So it is called as PEP. PEP, you know, phosphoenol pyruvate. Case means carboxylase. So this is the enzyme that is used in the conversion of carbon dioxide in C4 plants to oxaloacetic acid. This is the first step, right? So your atmospheric carbon dioxide is coming, right? So your phosphoenol pyruvate is fixing. It forms the C4 acid. So C4 acid is your oxaloacetic acid. So all this is happening in the mesophyll cell. Yes? Okay. So this oxaloacetic acid, which is the first stable compound, now moves inside the inner layer, that is the bundle sheath layer. When it moves inside the bundle sheet layer, it gets converted into another C4 acid. So far this was happening in the mesophyll cell. Now what happens, this oxaloacetic acid, it travels to the bundle sheet cells. So when it travels to the bundle sheet cells, it changes into another isomer with a 4-carbon only. This is still a 4-carbon. So when it moves inside the bundle sheet cells, it changes into another 4-carbon compound, that is malic acid. So this malic acid is again a 4-carbon compound. So this C4 acid is the malic acid. So once this malic acid has come inside your mesophyll cell, what happens? This undergoes decarboxylation reaction. This undergoes decarboxylation reaction. Decarboxylation, which means removal of 1 carbon. So when it undergoes removal of 1 carbon, okay, yes, you're right. That is what is here, the same malic acid, okay. So when it removes 1 carbon dioxide, it forms a 3-carbon compound plus a carbon dioxide. So this 3-carbon compound is your pyruvic acid. So when the malic acid enters the bundle sheet cells, it undergoes decarboxylation. It forms pyruvic acid and your carbon dioxide. This carbon dioxide will now follow your C3 cycle. So same thing, again here the acceptor of acceptor of carbon dioxide will be RuBP because it is following the C3 cycle. Yes. Now this pyruvic acid goes back to the mesophyll cells so that there is regeneration of your PEP. Always the primary acceptor should come out. So in the C3 cycle, the primary acceptor is RuBP and here the primary acceptor is PEP. So the last step is that the acceptor should be regenerated. Alright, yes, clear. No doubt. So first it starts with the outer layer, that is mesophyll. So your carbon dioxide is accepted by a 3-carbon compound, that is PEP, phosphoenol pyruvate, which gets converted into oxaloacetic acid, which is a 4-carbon compound in the presence of pepcase. This oxaloacetic acid moves inside the bundle sheet cell and then it gets converted into the malic acid. Alright, so this malic acid undergoes decarboxylation reaction where it forms pyruvic acid plus carbon dioxide. This pyruvic acid goes back to the same location to regenerate the PEP, whereas this carbon dioxide undergoes your C3 cycle. So this is the main biosynthetic pathway. So now I hope you're able to correlate. Yes? Fine. So what is the energy consumed here? In C4 cycle, you have C3 cycle also happening. So in C3 cycle, we know how many ATP is consumed. In C3 cycle, so you have 3 ATP plus 2 NADH, right? So in the C4 cycle, you have extra 2 ATP getting consumed. So totally for 1 molecule of carbon dioxide, 3 plus 2, 5 ATP plus 2 NADPH are being used. So for 6 molecules, it is 30 ATP plus 12 NADPH that is being utilized. Clear? Yes. So points to remember. So C4 cycle is also known as Hatch and Slack pathway because these are the scientists who discovered it. Alright. So the primary acceptor is PEP, phosphoenol pyruvate, which is a 3-carbon compound. Right. The enzyme which is used in this conversion is pepcase, phosphoenol pyruvate carboxylase. Fine. The first stable product is a 4-carbon compound, that is the oxaloacetic acid. So where it happens? First in the mesophyll cells, next in the bundle sheet cells. So here the C3 cycle happens in the bundle sheath cells, and in the C3 plants it happens in the mesophyll cells. How many is the ATP or the energy that is being utilized? For 6 molecules of carbon dioxide, it is 30 ATP plus 12 NADPH. Clear? The first two photosynthetic pathways are clear, that is C3 and C4, right? So was this session very useful? Did you guys understand what you failed to understand before? So that is the entire purpose of bringing this highlight series, to highlight the important topics which you haven't understood, for which we have to spend time and understand, right? So we will stop here, and the next two pathways we will discuss in the next week. Alright, so if you genuinely like this session, so please don't forget to like, share and subscribe. I will meet you on Monday at 3:15. We are discussing a sample paper. We'll continue the discussion of the sample paper. I'll continue the highlight series on Tuesday and Thursday. Alright, so until then, this is me signing off. So have a fabulous weekend ahead. Thank you and take care. Bye.